Good morning, my name is Kaisei Gwajan Robert, and I'm student at INES in year 3 in Biomedical Laboratory Sciences. So I'm going to explain about the one of the procedures that we use in the applied immunology in diagnosis of many diseases. So at the end of this presentation, we are supposed to understand the principle for the agglutination reaction, for the principle for the lattice agglutination, the principle for the precipitation reaction, the principle of the radioimmunodiffusion test, and even the principle of ELISA test. So the first one, I'm going to explain the principle of agglutination reaction. The agglutination reaction is a process where the particles such as cells or beds come together due to the interaction between the antigens and antibodies. So this reaction is commonly used in the immunology and diagnostic tests to detect the presence of specific antigens or antibodies in the sample. What you have to know for this agglutination reaction is that when we are doing the test, that is, we are going to interpret the result by considering the agglutination reaction. What we observe here, or what we are going to test, is the clumping that happened after the antigens and antibodies they clump together to form the complex. So here is about the diagram that is going to explain the different techniques for the agglutination test. The first one is the direct comb test, and the second one is indirect comb test. For the direct comb test, you can see that we obtain the blood sample from patients, which is, for example, this one is having immunomediated hemolytic anemia. Means that the antibodies are shown attached to the antigens on the red blood cells. Then, after getting that serum from this patient, we are going to add the anti-human globulin to the washed patient sample that we have been obtained. Then what you are having here, that you are going to see that the red blood cells they agglutinate, then anti-human antibodies form link between red blood cells by binding to human antibodies in the red blood cells. So this is for the positive result for the direct comb test. The second one is for indirect comb test, where we are having the recipient serum that is obtained and it is containing the antibodies or immunoglobulins. And here this donor blood sample is added to the tube with the serum. Then after, when we are having the recipient's immunoglobulins that target the donor's red blood cells, they form antibody antigen complex. And then after, we add the anti-human globulin or the Combs antibodies to the solution. Then for the positive, we are going to see that the agglutination of red blood cells they occur because human immunoglobulins are attached to the red blood cells. And the difference between this direct comb test and the indirect comb test, as you can see, for the direct comb test, you are going to detect the presence of the antigen, the presence of antigens from the blood sample of the patients. While for this indirect comb test, we are going to detect the presence of antigens. The second diagram is about the agglutination in different blood types. For this first one, you can see as it's blood group B, means that there is agglutination when we add the anti-human globulin on the blood group A. As you can see, there is agglutination on A. When we see the agglutination on blood group A, we are going to see that the blood is blood group B. For the A, the agglutination happen on the blood type B. And for this blood group O, as you can see, there is agglutination in both A and B. As the reason is that for this, there is for those both A and B, they are having the antigens that are going to clump together to antibodies to form the complex. While for the last one, you can see that there is no agglutination. The reason why there is no agglutination in both A and B is that there is no antigens. That's why we are going to see, we used to see that the the patient with the blood type AB, they are universal recipient, means that they can take every type of blood from every patient. And while for this blood group O, they are universal donor, they can give the blood group to everyone. So here it's about the factors that affect agglutination. The first one is concentration of antigens and antibodies, environmental conditions like pH and temperature, and ionic strength. And the last one is nature of antigens. There are many factors that affect agglutination. Those are the main ones. So the second one is about the latex agglutination. So the latex agglutination is a diagnostic technique that uses latex pads coated with the specific antibodies or antigens to detect the presence of corresponding antigens or antibodies in the sample. For this latex agglutination, you are going to see the clamping or agglutination of these birds serves as the visual indicator of positive reaction. Here is a diagram that is going to explain about the latex agglutination. Here you can see that there is suspended latex particles coated with antibodies, and then we add analyte, then the antigens cause the close linking and aggregation. So the application for the latex agglutination, we are having the infectious disease diagnosis, where we detect the bacterial, viral, even fungal antigens or antibodies. The second one is blood group typing. The third one is rheumatoid arthritis, the hormone detection, and the last one is about the drug testing. Actually, there are many applications for this latex agglutination, but those one I'm explaining is about the main ones. So the third question is the principle for the precipitation reaction. The precipitation reaction is based on the interaction between soluble antigens and the corresponding antibodies to form the insoluble complex. And those complexes, they aggregate and precipitate out of solution when the antigens and antibodies are present in the optimal proportions. So this diagram is about the precipitation reaction. Here we are having the antigens and antibodies, and they're going to clump together to form the antigens antibody complex. The application, we are having the diagnostic techniques where we identify the specific antigens or antibodies in diseases like infections or autoimmune disorders like rheumatoid arthritis and even those immunodeficiency syndromes. The second one is protein quantification, where we measure the concentration of antigens and even disease monitoring by tracking the level of specific antibodies in chronic or infectious diseases. Then the next question is about how is radioimmunodiffusion test is performed. First of all, the radioimmunodiffusion test is a quantitative method that we measure the concentration of antigens in the sample by observing its interaction between the antigen antibodies which is embedded in a gel. The antigen diffuse radially to form a well into the antibody containing gel, forming a visible precipitin ring at the zone of equivalence. So the procedures for this immunodiffusion test, we have in the first one, we prepare a gel. While we are preparing a gel, we mix antibodies specific to the antigens with agarose gel. Then we pour onto a plate and allow it to solidify. Then we punch wells by creating a small and equidistant wells in a gel. Then we load antigens by adding test samples and known antigen standards into the well. We incubate. As you can see, we incubate at 37 degrees Celsius as it is the optimum temperature for the human. Then we measure the ring, the last one, we determine the concentration. So when we are determining the concentration, we compare a ring diameters to a standard curve prepared from the known antigen concentration to quantify the antigen in the test sample. So the last question is the principle of the ELISA technique. First of all, this ELISA technique is said to be the enzyme-linked immunosorbent assay. So this ELISA technique is based on the specific binding of the antigen to its corresponding antibodies. Combined with the use of an enzyme-linked detection system to produce the measurable signals. Here, when we are doing this ELISA, we determine the color change, the intensity of how the color has changed. The signal intensity correlates with the amount of antigens or antibodies in the sample. Then we enable qualitative or quantitative analysis. So lastly, this is the references for my presentation, and I try to get those references so that everyone who can search to get the sources of where I've got those things, you can use those references to get them. So thank you so much. Thank you for your attention, and thank you.